Welcome to the PubMed Daily Review for March 09th. Today we scanned 1353 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, 2-year outcomes following Delandistrogene Moxeparvovec treatment in ambulatory patients with Duchenne Muscular Dystrophy, Phase 3 EMBARK trial by Jerry R. Mendel and Louise R. Rodino-Klepak from Gene Therapy, Nationwide Children's Hospital, Columbus, Ohio, USA. Let's break down its key contributions. Duchenne Muscular Dystrophy, or DMD, is a devastating genetic disorder that progressively weakens muscles, leading to loss of ambulation and a shortened lifespan. Current treatments focus on managing symptoms. Delandistrogene Moxeparvovec represents a novel gene therapy approach designed to restore functional dystrophin. While initial 1-year data showed promise in safety and some functional aspects, the primary goal of statistically significant improvement wasn't met, prompting a closer look at longer-term effects. This study followed 64 ambulatory boys with DMD, aged 4 to under 8 years, who received a single dose of Delandistrogene Moxeparvovec. To assess long-term effects, their 2-year outcomes were compared against a carefully matched external control group of 143 patients receiving standard corticosteroid treatment. The primary focus was on changes in functional abilities, measured by the North Star Ambulatory Assessment and various timed function tests. After 2 years, the results were compelling. Patients treated with Delandistrogene Moxeparvovec showed statistically significant benefits in both the North Star Ambulatory Assessment and key timed function tests when compared to the external control group. This indicates a sustained stabilization or slowing of disease progression. Importantly, microdystrophin expression remained stable, and no new safety concerns emerged during this extended follow-up period. This 2-year data suggests that Delandistrogene Moxeparvovec offers a sustained benefit in maintaining functional abilities for boys with DMD. The use of a matched external control group was crucial for demonstrating these longer-term effects. While these findings are encouraging, continued long-term monitoring is essential to fully assess the therapy's durability and its ultimate impact on delaying the loss of ambulation. Up next, our second paper out of 5. Fragmentomic liquid biopsy enables early breast cancer detection, molecular subtyping and lymph node assessment by Yuxuan Zhu and Chao Ni from Breast Surgery, Second Affiliated Hospital, Zhejiang University School of Medicine, Hangzhou, China. Let's break down its key contributions. Breast cancer remains a major global health concern, and early detection is key to improving outcomes. However, current screening methods like mammography have limitations, particularly in certain populations and settings. Liquid biopsies, which analyze cell-free DNA in blood, offer a promising alternative. This study explores a novel fragmentomic approach to see if it can overcome existing challenges in breast cancer detection and management. This multi-center case control study enrolled over 790 participants, including patients with breast cancer and healthy controls. They analyzed cell-free DNA from blood samples using advanced fragmentomic profiling. A machine learning model, called TuFEst, was developed to analyze these fragmentomic patterns for detecting cancer, determining its subtype, and even predicting lymph node involvement. The results were very promising. The TuFEst model achieved an impressive 95% sensitivity and 78.3% specificity for detecting early breast cancer. It also showed high accuracy in identifying malignancies that were missed by conventional imaging. Furthermore, the model performed exceptionally well in external validation, indicating its robustness and potential for real-world application. This study highlights the significant potential of cfDNA fragmentomics as a versatile liquid biopsy tool for breast cancer. It could revolutionize management by enabling early detection, molecular subtyping, and lymph node assessment from a single blood test. While these findings are exciting, further research and clinical validation are crucial to translate this technology into standard clinical practice. Up next, our third paper out of 5. personalized ctDNA analysis for detection of residual disease and recurrence in surgically treated HNSCC patients by Suzanne Flack and Philip Baumeister from Otorhinolaryngology, Head and Neck Surgery, University Hospital, LMU Munich, Munich, Germany. Let's break down its key contributions. Head and neck cancers remain a significant challenge, with many patients experiencing early relapse. Unfortunately, current methods for monitoring recurrence after surgery aren't always effective. This study explores the potential of ctDNA, a marker found in the blood, to help us catch these recurrences much earlier. This study followed 76 patients who had surgery for head and neck cancer. They collected blood and saliva samples before and after surgery, and during follow-up. Using personalized ctDNA assays, they looked for tiny fragments of tumor DNA to see if it could predict or detect recurrence earlier than standard methods. The results were quite impressive. The ctDNA assay was able to detect recurrence in over 91% of patients who relapsed. Even more importantly, it often detected this recurrence a median of 119 days before it was clinically apparent. Preoperative ctDNA was also detected in a high percentage of patients, suggesting its potential for early risk stratification. This study highlights the significant potential of ctDNA monitoring in head and neck cancer. By detecting recurrence earlier, we might be able to intervene sooner, potentially improving treatment success and patient survival. While promising, more research is needed to establish this as a standard part of patient care. Up next, our fourth paper out of 5, a KRAS G12V targeted bispecific T-cell engager promotes immunity against colorectal solid tumor by Nan Nguyen and Quang Thaching Nguyen. Let's break down its key contributions. KRAS mutations are found in a large percentage of cancers, making them a major focus for drug development. However, the KRAS G12V mutation has historically been very difficult to target directly. Recent breakthroughs in T-cell receptor technology offer a promising new avenue to harness the immune system to fight these challenging cancers. Researchers engineered bispecific T-cell engagers, called SIRs, designed to recognize the specific KRAS G12V mutation presented by HLA-A*11:01. These SIRs were tested in 2D and 3D in vitro models of colorectal cancer. The goal was to see if these engineered receptors could effectively direct T cells to kill tumor cells carrying the KRAS G12V mutation. The study found that the engineered SIRs, specifically TCER01 and TCER02, were highly effective. They successfully induced T-cell-mediated killing of colorectal tumor cells. Importantly, these SIRs demonstrated high specificity for the KRAS G12V peptide and showed minimal cross-reactivity with similar peptides, indicating a precise targeting mechanism. This research presents a promising new therapeutic strategy for KRAS G12V mutated colorectal cancer using T-cell engagers. While the specificity for a particular HLA type is noted, this work significantly advances the development of TCER-based immunotherapies. Future studies will likely focus on expanding this approach to other KRAS mutations and HLA types. Up next, our fifth paper out of 5, Microbial scFAs as Epigenetic Mediators, Fine-Tuning the Gut-Brain Axis in Neurodegenerative Disorders by Xiaotsui Su and Jingyu from A Department of Anesthesiology, Affiliated Hospital of Nantong University, Medical School of Nantong University, Nantong 226001, China. Let's break down its key contributions. Neurodegenerative diseases, such as Alzheimer's and Parkinson's, are devastating conditions affecting millions worldwide. While genetics play a role, a significant portion of disease risk remains unexplained. Emerging research highlights the gut-brain axis, the communication pathway between our gut and brain, as a key player. Specifically, short-chain fatty acids, or scFAs, produced by gut bacteria, are showing promise as crucial mediators in this complex relationship. This review delves into how scFAs, produced from dietary fiber fermentation, impact neurodegeneration. The research synthesizes findings from various studies, examining how scFAs, particularly butyrate, function as epigenetic regulators. Key mechanisms explored include their role as histone deacetylase inhibitors, their influence on DNA methylation, and emerging roles in histone lactylation, all of which can remodel gene expression in the brain. The review highlights several key findings. scFAs, especially butyrate, were shown to be potent inhibitors of histone deacetylases, leading to increased histone acetylation. They also modulate DNA methylation dynamics by influencing the balance of TET and DNMT enzymes. Furthermore, emerging evidence points to histone lactylation as a novel scFA-sensitive mechanism. These epigenetic changes are crucial because gut dysbiosis, often characterized by reduced scFA production, is linked to neurodegenerative diseases. In conclusion, scFAs represent a significant link between the gut microbiome and brain health, acting as critical epigenetic mediators in neurodegenerative disorders. Their ability to fine-tune gene expression in the brain opens exciting avenues for therapeutic interventions. Strategies like dietary modifications to boost fiber intake or the use of probiotics to increase scFA-producing bacteria hold promise for preventing or treating these debilitating conditions. However, more research is essential to fully understand these complex mechanisms and translate them into effective clinical applications.